Well, we had a little change of schedule, but I'm really honored to be here with Amalio. He's a very distinguished geneticist and expert in genomics. And I thought, this is a general audience, we'd start with just talking a little bit about what is genomics, where has it come from? You know, we saw a little bit of the history. I mean, in our lifetime, we went from dreaming of sequencing the human genome to it's somewhat routine. Maybe you can give us some perspective.
Yeah, I think everybody knows Mendel, the monk that was checking the color of peas. No? Yeah. And that's about 150 years ago. Genomics went through a dark ages with the eugenetics. That was a low moment for the field, and then started mostly with rare disorders. And it became mostly the field of pediatrics. I think only in the last 15 years, genetics became genomics. And the problem of occasional variants in the human genome became the problem of data. And we've discussed about genomics as purely data science.
Yes, and I think it's important, again, for this audience to understand it's not that we've gotten smarter, as usual, and I think many of you are from the IT fields. Our technologies have gotten much better, and so our ability to probe questions have gotten better. Maybe you can give some perspective, because it said that human longevity is the largest DNA sequencing facility, which I didn't know. So,
okay, so let's give some numbers. For those that are only excited by numbers, okay, it's 6 billion data points per person. And this is on the basic code, that's the software. Of those, each one of you are 4 million nucleotides or differences from your next neighbor in the chair. And I think what we are learning today is that we have a tremendous amount of uniqueness. And the bad news is that this uniqueness is lottery tickets. So you have 9,000 lottery tickets of basically your own personal experiment of making it through life or just dying of some horrendous, terrible disease. So you have 9,000, 9,000 chances. It's not bad.
it's not bad.
Not bad.
It's not bad.
To win some award. One example that it comes to mind, when there's the flu epidemic, at the end of the year, the CDC is going to say it was a great year, not many people died. Yeah? And the ones that died are always the same, the old people, etc. But when you look at the numbers, every year, there's the equivalent of 3 plane loads of children that die.
Yes.
And they were healthy. So we reassessed that question. Well, are they healthy? I mean, the virus was the one for everybody had the same virus. So what's wrong that a healthy child will die? And we checked the lottery experiment. Did they get the wrong lottery ticket? And the answer is yes. So they experimented by a random error. We make errors each time we divide. They were playing with a random error, and they broke a gene. And they were born with a broken gene.
And I think it might be important to state to everyone, I think we're all very used to, when we hear about genetic diseases, we think about childhood diseases, birth defects, mental retardation, muscular dystrophy, cystic fibrosis. These are the most common genetic diseases that could be in a few % of the population. Now what we're talking about are much more complicated diseases where the mutations are much more hidden in a large population. So they've been a lot harder to sift out.
Yep.
And I think that's what's so fascinating about what you're doing in human longevity is you're turning this into an information problem.
So I think, particularly for you as the audience, this is data. It's an IT problem. It's a mathematical problem. And it's less and less and less actually a medical problem in the sense of the type of records that physicians use or biology. So it is a huge mathematical problem of frequency. We were talking about the building blocks of the genome are all, it's a game of mathematical combinations of variants that are actually moving like Lego blocks. So that's actually to encourage you to see this not as a biomedical problem, but actually it's a problem of basic sciences, of numerical sciences.
And we were talking in the back room, and maybe it's provocative, but it's been very striking to me, living in Silicon Valley and in this environment of innovation and IT and the power of CS, just how little innovation there's been around using this genomic information. I mean, obviously there's a lot of talk about it, but really the full effort and power of Silicon Valley I don't think has been brought to bear on this problem. What do you feel?
I mean, there's a very important point you made, is sequence the genome is it's a commodity. So we produce a genome every 19 minutes. So that's not the problem. The price tag is not the problem. It's about buying a couple of iPads. So that's not the problem. The problem is that when you finish that works, actually nobody does anything with it.
Yeah.
I give you some metrics of this thing because I think it's an eye-opener. So with 30,000 people so far, we can count it 500 million diversity examples. So there's 500 million number, which basically doubles, triples all the knowledge of the last 30 years of medicine, which actually goes by a factor of 500, all the knowledge on changes in proteins, coding proteins. Yeah. And if I gave you the number 500 million, the amount of genetic variants that we have been able to pinpoint with certain knowledge is 20,000. So meaning you can say like, well, those are the important ones, or you can say our market and our thing is the 400 million remaining variants.
So, to translate to this audience maybe is that we're all very similar to one another. We may not think we are, but we're extremely similar to one another. The difference is there are many rare changes that are occurring, and how those link to human health. Would you say that's the challenge now?
That is the challenge. That is let's put it under the frame of longevity, which is another very exciting thing. There's two longevities. One is the longevity of the species. If I ask you how long does a dog live, you're going to answer 16 years. If you ask how long does a human live, I hope you will answer 85.
Well, a lot of times when people ask how long should Jody live, they usually say not soon enough. That's a different story.
Okay. So, but I'm going to come to you.
Okay.
But as a species, you are estimating the species so far.
I'm the representative. Yes. That's a bad,
as a representative
bad shape.
Species, you are going to live to 85. Mm-hmm. But actually, people start dying at 60. So between 60 and 85, that's the tail, and that tail is Jody. Are you going to be in that part of the tail? And that tail is beginning to be understood more as genetic problems. Mm-hmm. So there is the genetics of being a long-living species and not a dog.
Yep.
Mm-hmm. And there is the mechanisms and the drug market and the drug discovery for making a human live like a turtle or whatever, or if it's your enemy, making a human live like a dog. Yes. You know? And that's some genetics, that's some drug development, that's some critical biology. And then there is the biology of Jody, of you, that is very different. Is which genes do you have by bad luck broken or in a version that is of not very good quality that will make you die at 60.
And the challenge of this, I think, has always been, and this has been the promise and the failure, I would say, of genomics is once we understand the genome or genomes, we're going to create therapies to cure diseases, right? That's the facile statement that's often made. Can you speak a little bit? Obviously, there have been successes, there have been failures, there's been false promises. There's a lot of hope. You know, how do we now go to the next level of acting on our genome and that genomic information?
So again, genome is software.
Yeah.
You know? So if you identify the faulty thing, you can predict the problem of the performance of the machine. So the first use is not treatment. It's not drug. It's actually prediction.
Yeah.
So yes, you have a reasonable chance to develop this cancer or this inflammatory disorder. And then it is whatever. It could be lifestyle, it could be drugs, it could be gene therapy. Depends how the planet evolves. And that will be early on. It could be pure drug development. So we can plot now on a protein, we can plot everything that we ever seen, and you will see that perfectly well defines the critical parts. Hands off. I don't do any biology. I just can tell you that's a part of the protein.
And a drug company would run off and
if you want to target, those are the critical things. Those are the flexible. So it is just data-driven understanding of what is possible and not possible. So you have those trajectories.
And there's obviously been some great triumphs in cancers with personalized therapies, you know, that are directed towards certain types of mutations that were pulled out from the Genome Project. So, you know, there has been amazing progress is made, but we still have a lot to learn. One thing I wanted to touch on a little bit is, you know, we often state that genomic information will be part of modern medical care, and you're also an MD. Genomic information is very modern and very digital. Traditional medical information, going back to the reflex text, is not very digital and modern. How are these pieces of information going to be merged together? How do you see that moving forward in the future of getting a consolidated view of a patient's health digitally?
So, first point, don't take a fax machine away from a doctor. He will have a crisis. So that's where we stand. Okay? Second, if you want to know who knows today where things are going, is the patient who comes and has already read Google. Yeah. And actually tells the doctor, you know, what about this, what about that, what about these things, that's my risk. Yeah. So one sad aspect of medicine is this becoming out of sync with society. So it does not work on numerical sciences, that's for sure. It works on opinion, it works on perception, it works on reductionistic approaches. Now, if you want to know whether a baby is doing well or not, you do something called an Apgar score that goes from 0 to 10. Okay. And basically it's just, does he look good? Is he breathing? Yeah. You know, you can do better. Another example that you may understand, if you walk today into an intensive care unit, and well, we have a good chance that we'll all end there. Me at 85, you at 60.
Soon, very soon.
So if you
the broken leg.
If you walk into an intensive care unit with your IT eyes, you will see that everybody is connected to machines. And they are collecting, I don't know, 1,000, 10,000, 50,000 data points per day. The decisions are taken by the opinion of thinking like it looks good and it may make it out. While actually you could go and plot all this data in a vectorial space and say, where is this person? Say, oh, that person is that corner. Who are all the other ones in that corner? All the guys who made it out of the intensive care unit. So nobody uses, I think there are some startups, nobody is using the data that actually is produced. I'm not talking about whether we know how to use it, that is not used. Yeah. That data is thrown in the garbage and it will not be used either for care or for development or for analytics of how this place works.
And I think part of the challenge is, and I feel this myself as an old-fashioned basic scientist that was trained on doing singular experiments and hypothesis-driven research, is none of us are trained really to view things through this kind of statistical mathematical eyes. And certainly the medical profession is a very tradition-bound profession. And so I think a lot of this will be also changing the way we train our doctors to use data and be savvy in the language of genomics.
I mean, you could make a case that if we got X% of the medical year coming from a master's degree on computer sciences, statistics, algebra, we could create that generation. But you would probably have to give them a double training.
So let's look into the crystal ball down like 1, 2, 3 years from now. I have 30,000 genomes. A few years from now, maybe millions of genomes. How is that information going to be housed? How is it going to be accessed? How is it going to be actionable? And then, second question, what about issues of privacy? And these are big bioethical issues, and I'm sure you've thought about these.
I'm coming from Switzerland.
Yes.
So for me, the genome is the same thing as the bank account.
Yes. Yeah. Yeah.
The tools have all been developed.
Yeah. That's beautiful.
Yeah.
God bless the Swiss.
No, so this is a thing. So one genome is in a simple format is around 150 megas. The raw format is very heavy. We just checked a particular experiment. We just looked through 1 petabyte of data to look at particular aspects. So it is big amount of data, needs progress in compression, needs progress in cloud. I mean, most of the things are now moving into cloud. It needs everything that accelerates. Some of the main tools today take 3 days to compute a genome. New versions take a few hours. So everything is going to be incremental on reducing the volume and traceability. Once that you have this pile of data that so far for us is not tenable, so we are now at multiple petabytes on 30,000 and spending too much money per month, so cannot scale economically, that will have to be solved. There's no way to stock today 1 million genomes easily, and even worse, to compute at affordable cost. Now, once that you solve those problems, and I put them as problems, can you protect? Okay, so you need 75 variants to have a barcode of anybody.
So a personal identifier.
So it's no problem. I work with some excellent colleagues that I call hackers, and they say that not that they are serious university people, but they are hackers at heart, and they have mapped in front of me somebody who exposed his genome in one place, map it to their Facebook, map them to all their social networks, pull their families, and we estimated the risk of disease on their family members.
So you've been able to do a family tree by hacking.
By hacking. So now, are there solutions? Yes, because we're not using Swiss bank accounting.
Yeah.
Okay. So what do we try? Cryptography, it works. Heavy computer footprint, so it has to improve.
Yeah.
It works. We can do not full, we can do many operations, and we start even doing COCs, etc., models on encrypted data where I don't know what I'm doing, but it's answering correctly. We have deployed encrypted genomics for clinics, where the doctor asks a question, the computer answers it correctly because he has the half public key or whatever, and he has never seen the data. And you can only see the data you are allowed to and the question that you are allowed to ask. So there are solutions that have always been there. There's now new generation solutions of coming, let's say, from phone systems, obfuscation, some noise here, some noise there. So there's a full field of security, and it's either not implemented because of cross-field lack of understanding, or not.
That's where this audience comes
Yes.
into play.
Or not implemented because we have not improved the computing properties that makes it affordable. But on reality, yeah, we can encrypt your genome, and that's it. That's not encrypt the trajectory of the genome. Encrypt your genome, and the operations of the genome, questions, comparisons, statistics, association analysis are on encrypted data. There is multi-party computing where we have tested 3 continents asking the same thing on one particular disease. They never exposed their individuals to the other centers, but we still computed together. And so just to tell you, yes, but it's all solutions in your field. It's not in my field, unfortunately.
Yeah.
I'm just pumping out genomes, and then they pile up.
We've been talking about our genome, but we are not alone. Can you maybe comment a little bit about the interplay of our genome and health with the other organisms that are floating around? I know you've done a lot of work in this sector.
So there is multiple genomes in you. No? So you have your microbiome. I can mention it. You have your viriome in your blood. So your blood actually is not so clean as you think, unfortunately. So we are understanding the parts that actually make this confrontation of the species.
And the standard eye-opening comment, which always fluctuates, is there's at least as many foreign cells in your body as your own cells.
Yes, and for those that want to feel human, actually 40% of you is actually extinct viral presence. And so you are made out of leftovers.
Leftover viruses.
Of viruses in your genome. That is what is called endogenous retroviruses. If you thought that the protein coding part of the genome was what was human, that is false, that's 2%. So actually you are more virus than you are human in that sense.
It's probably the biggest surprise of the human genome when it was first sequenced was just how few functional protein genes there were. We thought there'd be a lot more from estimates. So
I mean, there's a big apparatus. No, it's a small apparatus to make proteins and a big apparatus of regulation, almost like the federal government. So few people work, but lots of people regulate.
Yes.
So basically,
government model
the genome is a bureaucratic unit.
Yeah.
Yes.
But the implications of the microbiome beyond just an interesting curiosity for human health has certainly generated a lot of popular insights. You know, what are your feelings about this? And maybe circle back to longevity.
Yeah. So So we are an ecosystem. Like every ecosystem, there's been good examples, worse examples of disruption of the ecosystem. For the gut, it's called dysbiotic. And then the yogurt company, they will try to sell you something so you can get a eubiotic microbiome. There's been a lot of controversy. They say, well, if you get the microbiome of an obese mouse and you give it to a lean mouse, that lean mouse becomes obese, and vice versa. Yeah. So this brought, of course, huge attention about the possibilities for manipulation of the microbiome. There's been a shocking report that went not completely open. It was in Science, so it was big, but the splash lasted very little. Is that when you drink light Coke or light Pepsi or light whatever, actually it damages your microbiome to an important degree. And that damage was translated in that paper as actually being pro-obesity. So that got many of the drinking companies extremely nervous, and they actually stopped research.
Traditional.
Because if they see it, they are putting the liability. Yeah. And this, again, is one of these things that was brought up. It's still completely unclear how much of this is going to pan out to be true. But if these are true, I mean, we're talking about true perturbations of of our normal health by a number of things that are common, the type of drinks, the type of...
And I think it's very important to mention, this is a field that didn't really exist. We didn't ever talked about the microbiome 20 years ago because we didn't have the ability to measure it. And so once we're able to see something, all of a sudden it becomes important. It's kind of a standard theme in science.
But it's always failing in numerical sciences. So it gets back to you. Either it's big, it is not movable, either it's not well protected, or it's not treated as a mathematical quiz. That's what it is.
Well, you know, I'm a terrible question asker, and I'm sure there's much better experts. I imagine there's some questions, and yeah.
I would be curious to know your opinion about the critics on bioethics when it comes to improving the human genome to the point that we maybe affect evolution of the species as a whole.
Yeah. So bioethics, it's a tough one. So we should not go too much there because it goes with ideology, it goes to the position of where we want to go. We do manipulate human life profoundly with many of our actions. So it's not that like we are not doing good and terrible things to our species. There is a lot of gene therapy that does not touch the inherited part. So I can always say, we don't need to touch your fundamental genome. We are going to manipulate your what is called the somatic. That means the circulating cells already with their own things and modify. So if we want to push gene therapy, we can't do it without modifying the species patrimony. Now, I can turn the question, it's about an ethical question. If you were a parent of a child with a problem, wouldn't you want to correct it for him and for her and forever? And that's a different thing. So should we, if it were simple, and now there's tools that are becoming slowly more applicable, would you correct it or not? If you were HIV infected, and you know, because we have done it, it works, that we can change your cells and close the portal of entry, which is a receptor. It can get knocked out, the virus stops entering, and then that's it, you are done, you are cured. And it's been done, it works. Right now it's too tough, it's bone marrow transplantation, so it's not a light-hearted, but it can be done. Wouldn't you do it at your personal level? You would do it, I mean, species-wise. Well, I mean, sometimes you are repairing, so actually just putting it back where it should be, you are not changing it in a way that has never existed. So if you have a gene that is defective, all you are doing is put it back to the normal form. So
a lot of questions.
You can turn it around. Yeah.
How do you do disease association on scale? I mean, I assume one of the big problems in genomics right now is still the phenotype, like genotype-phenotype gap. So I'm curious, like, not just limited to monogenic diseases, but also more complex diseases, how you like sort of plan on, addressing this? Like, is it learning algorithms, things like that?
So there are many, these are super, so many answers. I'm going to start with the one that is the most funny. If you want, so much of the genome today, again, is rare. So statistics get into some trouble. There are tricks, there are ways to compound signals, fine. But as we discussed just before we walked in, there is one solution to solve the genome. It's crowdsourcing. So if you have 5 people in the USA that share the same variant that you have not seen anywhere else, and 5 people elsewhere, the solution is how to make these 10 people talk and write. And actually, we have implemented this thing. So we're implementing a search tool that you can search your own genome and other genomes. And we actually, the first thing we put was a window for crowd discussion. Because what we hope is that you will write and say, oh, I'm the only one that has this variant in the planet. Hallelujah. But you may be 1.20 meters. So you wonder whether that's why. So what you want to know is that the next person is going to write and say, I'm 1.20 meters, 2. That's it. The variant has been solved. So at the highest level, crowdsourcing is a solution to a problem that is a crowd problem. And when we already have 500 billion variants, I don't know exactly how I'm going to give a name to each one of them and a function, except if there is a massive level. At the other levels, there's a lot of conventional statistics. There's a lot of machine learning approaches. There is not a surprise there. Where there is a surprise is that the genome data is a perfect data string. And the other data people are sending to us is data junk. They give us interpretive things, opinion, thresholds, snippets of data. If I want to say I want to see a genome and the magnetic resonance, the image of the person, both are numerical problems. That's a string, that's a genome, it's fine, and I can deploy wherever tools. The other is a report from a radiologist. There's nothing I can do with a report from a radiologist. I can say whether the genome fits the report of a radiologist, but that's about it. I have no way to look at the structure of the data.
Merging the data is a big challenge.
So for me, the next challenge, and we were bitching about the medical records world. Medical records is about pushing Excel tables through the planet. And that's not data. That's distributed garbage.
Yeah.
So what we need is people who just pipes raw data as medical records for appropriate analytics. And then if it fits wherever they look like and they think about, fine. One very simple example before we go to the next question. You take most of the human diseases today, and they are not one disease. They are the name that somebody gave to it. And then they say, oh, I treat this cancer, and there's 50% of response. Just as well they have 100% success of one cancer and a 0 success on the other cancer. What do I know?
Yeah.
I have no idea. Because they are talking Chinese, or Hebrew, or Spanish, whatever.
Or Italian.
Italian. So if we want to deal with data, the first thing we have to agree is that it has to be raw data and not interpreted data. And I think in medical records, we are already gone in the wrong way.
Maybe a couple more questions? Yeah, over here. Sorry.
Hi.
Last one. I'm getting cut off.
Oh, okay. I hope I make it a good one. We're learning a little bit more about epigenetics and the role that it plays in disease and therapeutics and even how we function. And that's not even to speak of like siRNA or RNAi, things like that. Can you speak a little bit about how that will change data mining for genome expression data away from, let's say, like a central dogma-driven paradigm?
Yeah. No, very good, very difficult question. So in a pragmatic level, let's solve the basic code. How much does it contribute? There is something called the twins studies. So when you want to measure how much a gene determines a trait, you go to the twins. And the twins give you the precise, we believe, estimation. And then you can say diabetes is 50% inherited, and 50% is whatever else. So epigenetics actually is on the boundary, and we don't know actually it's coded, because epigenetics, after all, has to sit on actual nucleotides. And we don't know how much of it is environmental. So it's sitting in between. And I guess some of it is accounted for on what we think is hard-coded software. And half of it is being manipulated by the environment. And it's dynamic. Like many other things are dynamic. Folding of the genome is dynamic. So if you take the genome at 8, 7 in the morning when you wake up, you will have some loops interacting and kicking in with some transcription, and that is relaxed at noon. So those are all layers of code, perhaps, 3-dimensional code, etc. And I don't know when we're going to get to those.
This is the challenge of biology. I mean, we're making discoveries, every year regarding mechanisms that are fundamental to life that we didn't even know existed before. And so my belief is that, yes, it's all encoded in the genome, but boy, we have a lot of work to do to then go to you sitting in the chair. And I think that's going to be the challenge. But there's no question that genomics and the type of tools that you're developing at human longevity, except as applied to me, are really going to make a difference in the world. And I really want to thank you for
Okay, thank you.
a wonderful discussion.